Tonight's lecture is over gene expression. In a multicellular organism, every somatic cell, that is, every cell except the gametes, is produced by repeated rounds of mitosis starting from a zygote. Since a cell's genes are duplicated with each round of the cell cycle, each somatic cell has the same DNA as the zygote. Simply put, every somatic cell contains every gene. If every somatic cell contains identical genetic instructions, how do cells become different from one another? Cells with the same genetic information can develop into cells with different structures and functions only if gene activity is regulated. Control mechanisms must turn on certain genes while other genes remain turned off in a particular cell. In other words, individual cells must undergo cell differentiation, that is, they must become specialized in structure and function. It is gene regulation, the turning on and off of genes, that leads to this specialization. So what does it mean to say that genes are active or inactive, turned on or turned off? As we discussed in the previous chapter, genes determine the nucleotide sequence of specific mRNA molecules, and mRNA in turn determines the sequence of amino acids and proteins, that is, DNA to RNA to protein, the central dogma of molecular biology. A gene that is turned on is being transcribed into mRNA, and that message is being translated into specific proteins. The overall process by which genetic information flows from genes to proteins, that is, from genotype to phenotype, is called gene expression. Because all the differentiated cells in an individual organism contain the same genes, the differences among cells must result from the selective expression of genes, that is, from the pattern of genes turned on in a given cell at a given time. Such regulation of gene expression plays a central role in the development of a unicellular zygote into a multicellular organism. During embryonic growth, groups of cells follow diverging developmental pathways, and each group becomes a particular kind of tissue. In the mature organism, each cell type, nerve or pancreas for instance, has a different pattern of turned-on genes. Moving on, how do cells decide to turn on which genes? Now there's a tricky question. Different cell types express different sets of genes. However, two different cells of the same type may also have different gene expression patterns depending on their environment and internal state. Broadly speaking, we can say that a cell's gene expression pattern is determined by the information both inside and outside of the cell. Some examples of information from inside the cell include the proteins that it inherited from its mother cell, whether the DNA is damaged, and how much ATP it has. And some examples of information from outside of the cell include chemical signals from other cells, mechanical signals from the extracellular matrix, and nutrient levels. But how do these cues help, quote-unquote, decide what genes to express? Cells don't make decisions in the sense that you or I would. Instead, they have molecular pathways that convert information, such as the binding of a chemical signal to its receptor, into a change in gene expression. As an example, let's consider how cells respond to growth factors. A growth factor is a chemical signal from a neighboring cell that instructs a target cell to grow and divide. We could say that the cell, quote-unquote, notices the growth factor and, quote-unquote, decides to divide, but how do these processes actually occur? Well, that is what this cartoon on your slide is trying to describe to you. The cell detects the growth factor through the physical binding of the growth factor to a receptor protein on the cell surface. The binding of the growth factor causes the receptor to change shape, triggering a series of chemical events in the cell that activate proteins called transcription factors. The transcription factors bind to certain sequences of DNA in the nucleus and cause transcription of cell division-related genes. The products of these genes are various types of proteins that make the cell divide, that is, to drive the cell growth and push the cell forward into the cell cycle. This is just one example of how a cell can convert a source of information into a change in gene expression. There are many others, and understanding the logic of regulation is an area of ongoing research in biology today. Moving on, to understand how a cell can regulate gene expression, consider the relatively simple case of bacteria. In the course of their lives, bacteria must regulate their genes in response to environmental changes. For example, when a nutrient is plentiful, bacteria do not squander resources to make it from scratch. Bacterial cells that can conserve resources and energy have a survival advantage over cells that are unable to do so. Thus, natural selection has favored bacteria that express only the genes whose products are needed by the cell. Imagine an E. coli bacterium living in your intestines. It will be bathed in various nutrients depending on what you eat. If you drink a milkshake, for example, there will be a sudden rush of the sugar lactose. In response, E. coli will express 3 genes for the enzymes that enable the bacterium to absorb and digest this sugar. After the lactose is gone, E. coli turns the genes off. It does not waste its energy continuing to produce these enzymes. Thus, a bacterium can adjust its gene expression to changes in the environment. But how does the presence or absence of lactose influence the activity of genes that code for lactose enzymes? Well, we see the answer to that question in this next slide. The key is the way that the 3 genes are organized. They are adjacent in the DNA and regulated, that is, turned on and off as a single unit. This regulation is achieved through control sequences, that is, short stretches of DNA that help turn on all 3 genes and turn them off. This coordinates their expression. Such a cluster of genes with related functions along with the control sequences is called an operon. The operon was first described in the 1960s by the French biologists Jacob and Monod. The cartoon on your slide describes a generic operon. How do DNA control sequences turn genes on or off? One control sequence, called a promoter, is the site where the enzyme RNAP attaches and initiates transcription. In this cartoon on our slide, we see that in between the promoter and the structural genes is something called an operator. The operator acts as a switch that is turned on or off depending on whether a specific protein is bound there. The operator and protein together determine whether RNAP can attach to the promoter and start transcribing the genes. So moving on, remember in our example of drinking a milkshake and the E. coli in your gut turning on the genes to metabolize lactose, this is a case where the lac operon is switched on and all the enzymes needed to metabolize lactose are made at once. So when there is no lactose around, the lac operon is in the off mode. So transcription is turned off because a protein called a repressor binds to the operator and physically blocks the attachment of the RNAP to the promoter. However, when there is lactose present, the operon is in the on mode. The lactose interferes with the attachment of the lac repressor to the operator by binding the repressor and changing the repressor's shape. In its new shape, the repressor cannot bind to the operator, and the operator switch remains on. RNAP is no longer blocked, so it can now bind to the promoter, and from there transcribe the genes for the lactose enzymes into mRNA. Translation produces all 3 lactose enzymes. Many operons have been identified in bacteria. Some are quite similar to the lac operon, while others have somewhat different mechanisms of control. For example, operons that control amino acid synthesis cause bacteria to stop making these molecules when they are already present in the environment, and this saves material and energy for the cells. In these cases, the amino acid activates the repressor. Armed with a variety of operons, E. coli and other prokaryotes can thrive in frequently changing environments. Moving on, eukaryotes, especially multicellular ones, have more sophisticated mechanisms than bacteria for regulating the expression of their genes. This is not surprising because a prokaryote, being a single cell, does not require the elaborate regulation of gene expression that leads to cell specialization in multicellular organisms. The pathway from gene to functioning protein in eukaryotic cells is a long one, providing a number of points where the process can be regulated, that is, turned on or turned off, sped up or slowed down. Picture the series of pipes that carry water from your local water supply to the faucet in your home. At various points, valves control the flow of water. We use this analogy to illustrate the flow of genetic information from a eukaryotic chromosome, a reservoir of genetic information, to an active protein that has been made in the cell's cytoplasm. The multiple mechanisms that control gene expression are analogous to the control valves in your water pipes. In eukaryotes, DNA is wound, folded, and compacted to fit inside the nucleus. The first level of organization, or packing, is the winding of DNA strands around the histone proteins. If DNA encoding a specific gene is to be transcribed into RNA, the nucleosomes surrounding that region of DNA can slide down the DNA to open that specific chromosomal region and allow for the transcriptional machinery, that is the RNAP, to initiate transcription. This type of regulation is called epigenetic regulation. Changes that occur to the histone proteins and DNA do not alter the nucleotide sequence and are not permanent. Moving on, transcriptional regulation in eukaryotes is complex, typically involving many proteins collectively called transcription factors. These act in concert to bind DNA sequences called enhancers and to the promoter. The DNA protein assembly promotes the binding of RNAP to the promoter. Genes coding for related enzymes, such as those in a metabolic pathway, may share a specific kind of enhancer or collection of enhancers, allowing these genes to be activated at the same time. In fact, repressor proteins that turn genes off are less common in eukaryotes than activator proteins that turn genes on by binding to the DNA. Activators act by making it easier for RNAP to bind to the promoter. The use of activators is efficient because a typical animal or plant cell needs to turn on, that is, transcribe, only a small percentage of its genes, those that are required for the cell's specialized structure and function. The quote-unquote default state for most genes in multicellular eukaryotes seems to be off, with the exception of housekeeping genes for routine activities such as glucose metabolism. Moving on, in some eukaryotic genes, there are regions that help increase or enhance transcription. These regions are called enhancers and are not necessarily close to the genes they enhance. They can be located upstream of a gene, within the coding region of the gene, downstream of a gene, or maybe thousands of nucleotides away. Enhancer regions are binding sequences or sites for transcription factors. When a DNA bending protein binds, the shape of the DNA changes. This shape change allows for the interaction of activators bound to the enhancers with the transcription factors bound to the promoter region and the RNAP. Like prokaryotic cells, eukaryotic cells also have mechanisms to prevent transcription. Transcriptional repressors can bind to the promoter or enhancer regions and block transcription. Like the transcriptional activators, repressors respond to external stimuli to prevent the binding of activating transcription factors. Moving on, within the eukaryotic cell, transcription occurs in the nucleus, where RNA transcripts are processed into mRNA before moving into the cytoplasm for translation by ribosomes. RNA processing includes the addition of a cap and tail, as well as the removal of any introns, that is, non-coding DNA segments that interrupt the genetic message. and it also includes the splicing together of the remaining exons. Moving on, sometimes a cell can carry out exon splicing in more than one way, generating different mRNA molecules from the same starting RNA molecule. With this sort of alternative RNA splicing, an organism can get more than one type of polypeptide from a single gene. Alternate RNA splicing is very common in humans. In one case, the RNA transcript from a gene can be spliced to encode 7 different versions of a cellular protein. Moving on, before the mRNA leaves the nucleus, it is given 2 protective caps that prevent the end of the strand from degrading during its journey. The 5' cap, which is placed on the 5' end of the mRNA, and a poly A tail, which is attached to the 3' end. Once the RNA is transported to the cytoplasm, the length of time that the RNA resides there can be controlled. Each RNA molecule has a defined lifespan and decays at a specific rate. This rate of decay is referred to as RNA stability. If the RNA is stable, it will be detected for longer periods of time in the cytoplasm. Also, the microRNAs are short single-stranded RNA molecules that are only 21 to 24 nucleotides in length. Like transcription factors and RBPs, mature microRNAs recognize a specific sequence and bind to the RNA to degrade the target mRNA. They rapidly destroy the RNA molecule. So let's quickly recap tonight's lecture. After tonight's lecture, you should be able to tell me why cells express different genes. You should also be able to list the key differences between prokaryotic and eukaryotic gene regulation. You should be able to describe the basic steps in prokaryotic gene regulation. You should know what epigenetic regulation is. You should know how transcription factors impact gene regulation. You should know what RNA splicing is. And you should be able to explain how RNA stability plays a role in gene regulation.